Welcome back to Dr. Liu's VCE Biology. Today's topic is determining the relatedness over time, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Cetaceans, whales, porpoises and dolphins are marine mammals belonging to the order Artiodactyla, even-toed hoofed mammals. The closest living relatives of cetaceans are hippopotamuses. Phylogenetic tree A summarizes the evolutionary relationships of 4 present-day cetacean species and the hippopotamus. The branch for Pakicetus does not extend to the present day, 0 Maya, which indicates that this species is extinct. A fossil named Ambulocetus was found in 1992 and dated at 49 million years old. Some paleontologists believe that it is a transitional fossil between the ancestral land mammal shown in phylogenetic tree A and present-day cetaceans. Predict 2 structural features of the Ambulocetus fossil that would provide evidence to support the hypothesis that it is a transitional fossil and suggest a survival advantage of each feature. Ambulocetus, believed to be a transitional fossil between the ancestral land mammal and modern cetaceans, would exhibit a combination of aquatic and terrestrial features: heavy bones for sinking and flat feet, or hooves for walking on land, paddle-like limbs or webbed feet for swimming and 4 walking legs for terrestrial movement, and nostrils and eyes on top of the head for surface breathing and vision paired with short legs suitable for walking in mud. The feature needed to be feasible in a fossil. The presence of lungs or gills was not feasible, as these would not fossilize. Sharks are marine fish of the order Chondrichthyes, cartilaginous fish. Phylogenetic tree B summarizes the evolutionary relationships of 3 present-day shark species and the main fish classes. Divergent evolution describes the splitting of 2 or more lineages from a common ancestral species, where populations accumulate distinct morphological or functional traits in response to varying environmental pressures, or ecological niches. The divergence of the bronze whaler and blacktip reef shark provides a clear case, as their shared ancestry 23 million years ago was followed by adaptation to different selective pressures. The evolutionary relationship between the hippopotamus and the killer whale, a cetacean, serves as a prominent example of divergent evolution. Although their most recent common mammal ancestor is thought to have lived approximately 55 million years ago, the 2 lineages have diverged dramatically as a result of contrasting selective pressures and adaptation to vastly different aquatic and terrestrial environments. Convergent evolution describes the independent process where unrelated organisms evolve similar traits or structures, analogous structures, typically in response to shared environmental pressures or ecological niches. This phenomenon is clearly demonstrated in the marine environment. The humpback whale, a mammal, and the whale shark, a fish, independently evolved streamlined bodies and specialized filter-feeding mechanisms, baleen versus gill rakers, to exploit pelagic plankton efficiently. Similarly, the predatory killer whale, a mammal, and the bronze whaler, a shark, both developed streamlined, hydrodynamic body plans and large dorsal fins for the speed, stability, and maneuverability required of an apex. The Australian scientists who identified the Burrunan dolphin, Tursiops australis, as a separate species in 2011 relied on multiple lines of evidence, including analyses from both living populations and museum specimens. The key findings were genetic data, such as mitochondrial DNA, mtDNA, and microsatellite, nuclear DNA, analyses, which demonstrated deep evolutionary divergence and reproductive isolation from the common bottlenose dolphin, T. truncatus, and the Indo-Pacific bottlenose dolphin, T. aduncus. This genetic evidence was supported by morphological evidence, which showed unique differences in cranial, skull measurements and a distinct tricoloration pattern, dark gray, light gray, and off-white, not found in other known bottlenose dolphin species. Interbreeding to test fertility. While the biological species concept defines a species as a group that can interbreed to produce fertile offspring, it is impossible and unethical to conduct this experiment on endangered wild dolphins. The genetic data confirming reproductive isolation, lack of gene flow, serves as the necessary evidence instead. The Burrunan dolphin, Tursiops australis, is a highly vulnerable species, with only about 150 individuals confined to two small, isolated populations in Victoria, Port Phillip Bay and the Gippsland Lakes. The Port Phillip Bay group is particularly isolated, experiencing little to no gene flow from the Gippsland Lakes. Given this isolation and the intense human impacts on Port Phillip Bay, including industry, recreation, fishing, and shipping, the Burrunan dolphin faces three potential future paths: divergent evolution, where differing selective pressures could lead to the Port Phillip Bay population evolving into a species separate from the Gippsland Lakes group; extinction, driven by the severe pressure from human activities on such a small, non-mixing population; or a population increase, which is only likely if dedicated conservation efforts are implemented to protect these genetically distinct groups from escalating anthropogenic threats. This presentation examined the powerful influence of selective pressures on species evolution. We began by observing convergent evolution, where distantly related organisms, such as the humpback whale, a mammal, and the whale shark, a fish, independently evolve similar solutions, like streamlined bodies and filter feeding strategies, to thrive in the same marine environment. We then shifted to the future of a species facing critical evolutionary challenges: the Burrunan dolphin, Tursiops australis. Identified as a distinct species in 2011 through rigorous genetic and morphological evidence, this dolphin is endemic to only two tiny, isolated populations in Victoria, Port Phillip Bay and the Gippsland Lakes. The Burrunan dolphin's small population size, $approx $150, and geographic isolation place it at a precarious crossroads, with two main outcomes driven by escalating human impact in Port Phillip Bay: speciation, divergent evolution, the strong isolation. Unique environmental pressures of Port Phillip Bay could cause this small population to diverge genetically from the Gippsland Lakes group, potentially leading to the evolution of a new, endemic species over time. Extinction, the immediate risk. Due to low numbers and chronic threats from human activity, pollution, vessel traffic, the Burrunan dolphin faces a high probability of extinction before significant evolutionary change can occur. The Burrunan dolphin stands as a stark reminder that while nature's pressures drive evolution, human pressures can drive species to the brink. Its survival is not guaranteed; it depends entirely on immediate and sustained conservation action. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.